Antibody is a glycoprotein, a globular glycoprotein. So it is called a globulin, immunoglobulin, and it is called gamma globulin as well. There are different names for antibody. So they are glycoproteins that are mainly produced by plasma cells, and when they are bound to B cell, they are called BCR. When they are secreted by plasma cells, they are called antibody. Let me skip this image. So, antibody, we have two identical heavy chain and two identical light chain. Okay? What do you know about the structure of antibody? Tell me anything you remember from your previous lectures or previous course. Light chain, two identical light chain, and two identical heavy chain.
Isolated bound. Yes.
Yes.
And Fab region.
They are disulfide bonds. Disulfide bonds.
Yes.
Okay.
Antigen binding site.
And where is antigen binding site? Yes. In each chain we have some domains. They are called domains. Okay, they are called domains. In light chain we have only two domains. They are generally called immunoglobulin domains. In the light chain we have two domains. In the heavy chain we have 4 and sometimes 5 domains. In the light chain we have one variable domain and one constant domain. This one is called variable because it's the site that binds to the antigen, and in different antigens it would be different because it should bind to different antigens. It's variable against different antigens. When we have many antibodies, indeed their variable region is different from each other, that they can bind to different antigens. Is that clear? For example, we have antigen A, B, C, D, E, F. And we have antibody that are binding to these antigens. So this variable region is different in different antibodies, that they are specific for different antigens. So they are called variable domains because they are different in for binding to different antigens. In different specific antibodies, we have different variable domains. Okay, is that clear? It's similar to the lock and key. Okay. To be better fit with the antigen that is going to bind to it. So it is called variable region. In the heavy chain again, we have one variable region. And those variable regions, when we have one variable region from light chain, another variable region from heavy chain, and those variable regions make a site for antigen binding, which is called antigen binding site. For example, this is antigen, this one is antigen, and this is the variable region of light chain, this one is the variable region of heavy chain. So now you can see that, for example, the shape of this antigen binding site is going to bind to this type of antigen, to this shape of the antigen. So its shape is in a way that can fit with them completely. But if the antigen is different, for example this one, and I'm another antibody, so the shape of the antigen binding site is different. Okay? So it is called variable region. For this epitope we need this for example, shape. For this epitope, we need another shape. So they are variable, they are different structures that enables the antibody to fit with different antigens or epitopes. Clear?
According to the shape of the epitope. According to the shape of epitope.
Yes. In order to bind to the shape, to different epitopes with different shapes.
What is the difference between heavy chain and light chain?
Light chain is a smaller chain, the heavy chain is the bigger chain, that's it. You know, the antigen binding site, okay, the antigen binding site is composed of one variable region of light chain and one variable region of heavy chain. And variable chain of light chain alone cannot bind to the antibody. And even variable domain of heavy chain by itself cannot bind. We need both variable regions in order to bind the antigen. Okay? Without both of them together make a site that can bind to the antigen. We need both of them. And the rest of the chain is called constant regions. So we have variable regions and then constant regions. In the light chain, we have just one constant domain or region, but in the heavy chain, we have 3 or 5 constant regions. So CS is constant of heavy, variable of heavy, variable of light, constant of light, and so on. Clear? So variable, two variable regions that they are together, they make antigen binding site. This part is called hinge. This part is called hinge. So antibody has 2 identical heavy chains and 2 identical light chains. So we have 2 antigen binding sites. Is that clear? We have 2 antigen binding sites. That both of them bind to one epitope. I mean one type of epitope. For example, if this one is against A, of course this one is against A because their chains are completely same. Okay? One antibody has 2 arms. So I am an antibody. This one arm is this one is another arm. 2 arms with 2 antigen binding sites. For example, if I am an antibody, I'm going to bind to those epitopes. So I have 2 arms. One of them for this part, another one for this part. And so both of them are completely same. Both antigen binding sites of an antibody, both of them are against just one epitope. Here, both of them are against A, both of them are against B, both of them are against C, and so on.
The variable domain can bind to antigen regardless its shape. Why do we need the constant one?
I'll tell you. So it's a very good question. That when we have these domains that they can bind to antigen, what is the function of the rest of the antibody? Why do we need the rest of the antibody? I'll tell you in the next, now after completing my explanation. So we have 2 antigen binding sites that both of them binds to one type of epitope. So antibody is called one antibody. This is one antibody. Indeed, it's a tetramer antibody. Tetramer means it's composed of 4 chains, 2 identical heavy chains and 2 identical light chains. So, it is that's why it is said that an antibody is monospecific. Monospecific means both of them are against one kind of epitope. For example, here epitope A, monospecific. It means that this arm is not against A, this one against B or other epitopes. Both of them against one epitope. So one antibody is monospecific. Okay? It's logical because the chains are completely same. The color is different here, but this domain is completely the same as this one, and this domain is completely the same as this one. So this shape is completely the same shape of this part. Okay? No question? No? So, I told you that one antibody has two arms, one arm here and another arm here. And in the part of the arms that they are bound to the rest of the antibody, it is called hinge. For example, this one is one arm, another one is one arm. So the hinge enables the antibody to increase and decrease the distance between its arms. For example, if the epitope is close together, it runs in this way to them. But if the epitope is far from each other, so the hinge allows the antibody to increase the distance between these arms. So the arms can move toward each other to increase or decrease the distance between them in order to enable the antibody to bind the epitopes with different distances on the antigen. Okay, so it is called hinge. So each arm of the antibody, which is composed of antigen binding site and one constant domain, it is called FAB, fragment of antigen binding. Fragment of antigen binding. Okay, so one antibody has two arms that each one is called Fab. So one antibody has two Fabs. Okay, the rest, this rest of the antibody is called FSC. It's just a name, FSC region. So what's the region of when this part of the antibody bind to the antigen in the antigen binding site? Why do we need the rest of the antibody? First of all, we need the rest of the antibody in order to stabilize the antigen binding site. Without the rest of the protein, it's a complete protein. If we cut protein from here, these variable domains are not stable. They will be degraded easily and they cannot bind even to their antigens. Okay? This is one function of the rest of the antibody, increasing the stability of the antibody. Another function is that those parts, antigen binding sites, have responsibility to recognize the antigen. But this part, which is called FC region, most of the function of antibody is done by the FC region. For example, you know that one function of antibody is that antibody is an opsonin. Do you remember opsonin? Opsonin, what is an opsonin? An opsonin is anything that facilitates phagocytosis. Okay? Is that clear or not? Do you remember? No? Opsonin is any substances, for example, this is a bacteria and I'm a macrophage. A macrophage can bind to the bacteria and phagocytose it. But an opsonin is something that binds to the antigen and make easier, facilitate the phagocytosis of the antigen. Indeed, it is said that opsonins make the antigen delicious for the macrophage or phagocytic cells. So opsonins are any substances, any materials that make the antigen delicious for the phagocytic cells, or they indeed facilitate phagocytosis. One function of antibody is that they are opsonin. They facilitate the phagocytosis. What does that mean? We have a bacteria and we have antibody against this bacteria. Now this antibody is going to facilitate phagocytosis, and we have a macrophages. This macrophage when bind to the antibody can phagocytose whole complex of antibody and bacteria. So here is the FSC, and here are the antigen binding sites. So macrophage, after binding of antibody to the bacteria, macrophage bind to the FSC of the antibody and phagocytose the bacterium. So binding of macrophage to this antibody is made by the FSC. Without the FSC, without this arm, this part, antibody won't be an opsony. Okay? Clear or not? It sounds not. Or yes? Not? I'll explain it to you. I have to use different colors, I think. So, I'm talking about one function of antibody. One function of antibody is opsonization, okay? Opsonization. Opsonization means making a particle, a bacteria, delicious for phagocytosis, or anything that facilitate phagocytosis. For example, we have a bacteria here, two bacteria. That one of them is coated by an opsony, for example, antibody. One of them is not coated by an antibody. And we have two macrophages. So this macrophage can phagocytose this bacteria, of course. But this macrophage can phagocytose this bacteria in a much potent way, it's much efficiently. And phagocytosis of bacteria will activate killing properties of macrophage. This macrophage, after phagocytosing this bacteria, will produce higher amount of cytokines, but this one lower amount of cytokines. The killing properties of this macrophage is weaker than this one because we have opsonization here. Here we do not have opsonization. It means opsonization, making phagocytosis easier and more efficient. Is that clear for the definition of opsony or not? Clear? So, now we have a bacteria and antibody against bacteria is produced by B cell. So they are the antigen binding sites. So when we draw the bacteria antibody in this way, this part is Fab, two Fabs, and this part is FSC. Fab indeed here is antigen binding site, two antigen binding sites. Is that clear? Is that clear for the structure of antibody? So we have two antigen binding sites. It's better to show light chains here. This one is better now. Light chains and heavy chains. Okay? Is that clear? So we have three antibodies that all of them are bound to bacteria by their antigen binding sites. They have the FSC part. This part is FSC, is free. So antibody is an opsony. So on the macrophages, we have some receptors that they can bind to those antibodies. So when those receptors, for which they are called FC receptors for FSC, they bind to FSC, FC receptors. So macrophages have FC receptors. After binding of macrophage by its FC receptor to the FSC of antibody, that this antibody is already bound to bacteria, so this bacteria will be phagocytosed very efficiently. Is that clear or not? So antibody is facilitating the phagocytosis of the bacteria. Without this FSC, we won't have any opsonization activity of the antibody. That's why I'm saying you the importance of FSC. Many functions of antibody, including phagocytosis facilitation or opsonization, is mediated by FSC, and some other functions that I'll tell you in the next slides. So antibody is a tetramer, two heavy chain, two light chain, antigen binding sites, two arms with same specificity, and a hinge that allow the arms to move toward each other in order to bind to epitopes with different distances, and one FSC, that in the FSC region we have some carbohydrate that are bound to the FC region. That's why we say antibody is a glycoprotein. What is the difference between glycoprotein and a protein? What is the difference between protein and glycoprotein?
Proteins only protein, like proteins, glycogen and protein.
Yes, glycoprotein is composed of protein and carbohydrates, not glycogen only, any carbohydrates. Okay, it's called glycoproteins are proteins that are bound to carbohydrates. Okay, glycoproteins, not only glycogen. Glycosylation means binding of carbohydrate to protein, not just glycogen or anything like glycogen. So any carbohydrate can bind to the protein. So antibody is a glycoprotein, and this glycosylation or binding of carbohydrate will increase the stability of the antibody as well. What does the stability means? It protects the antibody from degradation by different enzymes that we have in the interstitial tissue and body. Clear? No question? Antibody structure: FAB, FSC, antigen binding site, monospecificity, Hinge, FC functions. Clear? FC receptors. What is a FC receptor? What is FC receptor?
Increase ability.
Yes, you. What is a FC receptor?
Increase ability of the
FC
fibers.
receptors recognize FC part of antibodies.
Receptor for FC. Receptor for FSC of antibody. It binds to the FSC of antibody. That's it. We have different types of antibody, so we have different FC receptors. Okay? We have, I'll tell you in the next slide, we have IgG, IgA, IgM, IgE, IgD. You remember those types of antibody. So we have different types of antibody. We have different types of FC receptors on different cells. They are very important in the immune system, FC receptors. As the name shows, receptor for FSC. Okay? Receptors that just bind to the FSC of an antibody, a free antibody. No question? One function of antibody, let me give you another examples of the importance of the FC region. Until now, I have given you three importance of FC regions. One of them stability, another one opsonization, and this one. One function of antibody is activation of complement system. Do you remember complement system? The cascade of proteins. So, yes, I'll talk about them tomorrow. But complement proteins, antibody can activate complement. Antibody can activate complement. When one antibody binds to antigen, for example, then FSC can activate the complement system. So, without FSC, we won't have complement activation. That's why I'm saying you that most functions of antibody is mediated by FSC. Without FSC, we won't have those functions that I'll tell you some other functions of antibody in the next slides. So antibody, we have antibody in the blood, in the tissues, in the mucosal surfaces, but for example, in the mucosal surfaces, we have IgA antibody mainly, but in the blood, we have IgG antibody mainly, that I'll talk about them. So we have antibody almost in most part of the body, almost all part of the body, because they are plasma proteins that can migrate to the body, and they can exit the blood in the capillaries and go into the interstitial tissue. So they are called immunoglobulin domains as well, or regions. It's important to say that two heavy chains are bound together in the hinge region by some disulfide bonds, and light chains are bound to the heavy chains here. These disulfide bonds here, disulfide bonds, keep the integrity of the antibody, the structure of the antibody. Without these disulfide bonds, we won't have a complete antibody. In order to have a complete antibody, please, I know you are tired, but we have no other way. We will have 3 lectures each day. It would be tiring for you, I know, but there is no other way. You need to balance your energy for all lectures. Please have a look here and listen. It sounds you have a very important meeting over there, and even you cannot control your sound, your voice. Okay. Here is another shape for the antibody: variable domains, constant domains, hinge region, antigen binding site, and FSC, this part, and most biologic activity of antibody is done by FSC. You can see here, we have different types of heavy chain. We have different types of heavy chains. Mu chain, gamma, they are proteins with different names. Mu chain, gamma chain, alpha chain, delta chain, and epsilon. 5 different chains. They are different heavy chains. And based on these heavy chains, we have different types or isotypes of antibody. Mu is for IgM, mu is for IgM, gamma is for IgG, and delta is for IgD. Another one, alpha, is for IgA, and epsilon for IgE. So it means any antibody that its heavy chain is in mu, it is called IgM. Any antibody that its heavy chain, both of them, not one of them, we do not have 2 different heavy chains bound to each other. 2 heavy chains are same heavy chains. In the IgM, both of them are mu chains. In the IgG, both of them are gamma chains. In the, for example, IgA, both of them are alpha chains. I'm talking about different heavy chains, not light chains. The naming of isotype of antibody is just comes from the heavy chains, not light chains. Clear? Clear? Do you know the difference between the IgG, IgM, and IgD? They are different proteins or different types of heavy chains, and they are mainly for the constant regions. There is a tiny difference between them. And again, here you can see, again, disulfide bonds between hinge region, heavy chain, and light chain, variable of light, variable of heavy chain, and FC part, and hinge region. And FAB, this part is called FAB, and this part is called FSC. One other important function of FSC is solubility. This FC part makes the antibody more soluble. It enables the antibody to move easily throughout the body in the different fluids of the body. To light chains, to heavy chains, I talked about them. You do not memorize the details like this. So the number of amino acids of each domains. You do not know anything about this. I'm crossing out in order to not memorize them. effector function antigen binding site. This one is Fab, this one is FSC. There is no need to memorize them. There is no need to memorize these parts. So different heavy chains for different isotypes of classes of the antibody. So we have 5 different isotypes of different classes of antibody that their difference is only in the heavy chain protein. We have 2 different light chains that you do not need to memorize them. Sometimes we have some subclasses. What does that mean? And it means that, for example, we have 2 IgAs, IgA1 and IgA2, and 4 different IgGs as well, IgG1, 2, 3, and 4. You absolutely are going to face with this type of antibodies. You are going to be a physician, so all of them are being detected in the laboratory. Antibody is very important part of immunology because you are dealing with different antibodies in the laboratory. IgE, IgA, IgM are detected in different tests of the laboratory, clinical laboratory. I mean, not only research laboratory. Besides, antibodies are widely used in the treatment of different diseases. So antibody lecture and antibody function and structure is very important and useful for you, that you are going to be a physician, inshallah. So you can see here the different classes of antibody: IgG, IgA, IgM, IgE, IgG. And usually IgA that is in the mucosal surfaces, which is called secretory IgA. IgA that is found in the mucosal surfaces, it is called secretory, is usually a dimer IgA. And IgM is always a pentamer. Okay? IgM, we have IgM in this way, always a pentamer. Indeed, 5 single antibody is bound together with one chain. It doesn't matter the name of the chain. So it is the structure of IgM. So which of the antibodies are heavier than the rest of the antibody? IgG, IgA, IgM, and IgE. IgM is heavier. It's very important. It's very important. And IgM, I'll talk about the function of each classes. You do not need to memorize the content of this table. B cell receptor is indeed, what is a B cell receptor? What is the difference between antibody and a B cell receptor? What is the difference? Most of the questions I ask are indeed the questions of the exam. What is the difference between a BCR and an antibody?
The same.
They are the same, but there is a tiny difference between. It is important to know that they are the same proteins, but there is a tiny difference between them. Antibody is a free protein that is released by the plasma cells, but BCR is a B cell bound antibody. It's on the surface of B cell. It's the only difference between them. So you can see here, this is an antibody which is bound to the cell membrane. It is bound to the cell, and that's why it is called membrane immunoglobulin. This is another name for BCR, membrane immunoglobulin. I told you, immunoglobulin is another name for the antibody. So we have different kind of Fc receptors. I told you what is a Fc receptor. Fc receptor is a receptor for the efficacy of antibody, and many functions of antibody is mediated by these kind of Fc receptors that I'll talk about them one by one in in the next slides or next lectures. The function of antibody, opsonization, I told you. ADCC, I'll talk about about the ADCC in the next lectures. Until now, I have told you 2 times about ADCC. When I was talking about NK cells, and now I'm talking about antibody. So that's why it is called antibody dependent cellular cytotoxicity. This cytotoxicity is made by NK cells. So in the ADCC, we have both involvement of antibody and NK cells. And I'll talk about them in the next slides. So one function of antibody is ADCC. Opsonization, ADCC, 2 functions. Another one is the presence of antibody in the epithelium, in the surface barrier. I'll talk about IgA in the surface barrier, in the mucosal surfaces, completing the barrier immunology. There is a very important receptor for the antibody, which is called neonatal FC receptor. This is responsible for the transportation of maternal IgG to the fetus. From those classes of the antibody, just IgG can pass the placenta and go to the body of the fetus. So, for the transfer of IgG from the blood of the mother to the blood of the fetus through the placenta, we need one receptor which is called neonatal FC receptor. FC receptor of the fetus. Neonate is another name for the fetus. Okay? So it is called FC receptor, neonatal FC receptor. It is responsible for the transportation of IgG only, not IgM, not IgA, not other receptors to the blood of the fetus. Let me check my slides. I'm trying. The only things that I'll talk about the function of antibody in the next lectures, but that's enough for today. See you tomorrow at 8.